Welcome to the Cafe Culture podcast, a season of discussions on culture, politics, philosophy and science.
At a talk recorded on the 16th of May, 2016, Catharien Hilkens discussed cell therapies.
Thanks very much, Simon, for the introduction. Can everybody hear me? Yeah? So good evening, everyone. It is a great pleasure to be here tonight and to talk to you about cell therapies, a subject that occupies my thinking almost every day. So the use of therapeutic cells, or cells as a therapeutic tool, is currently undergoing a revolution in the field of medicine. Cell therapies have been developed over the last decade for the treatment of cancer, but also for complex inflammatory diseases that are often difficult to treat, like rheumatoid arthritis. Now, a number of these cell therapies have now undergone testing in clinical trials, and the first results are highly encouraging. I don't know if you heard the miracle stories, for instance, about so-called CAR T cells that can cause a complete remission in patients with an untreatable form of B-cell leukemia. And there's no doubt in my mind that cell therapy is here to stay. So the time is right, really, to start engaging and informing the public about cell therapies, and hence this talk tonight, and therefore I was invited to talk to you tonight. So today, what I would like to do is first of all discuss with you some general features of cell therapies. What is a cell therapy, and how does it differ from the conventional drug therapies that we know? Secondly, I would like to tell you our story. I was appointed at Newcastle University in 2003 to develop a cell therapy for the treatment of rheumatoid arthritis. And together with many team members, and some of them are here scattered throughout the audience, we recently successfully completed a phase 1 trial with our cell product, and we showed that it is safe. Now, we still have a very long way to go before our treatment can become part of clinical care, but the first important and time-consuming steps have been taken. Finally, in the last part of my talk, I would like to start discussing some of the difficulties that we are facing with bringing this new cell therapy to routine clinical care. So let me start. So since you're all attending a talk on cell therapies, my guess is that you have an interest in the field, or you may already have a pretty good idea what we mean by cell therapy. So can I ask members of the audience, what do you think is a cell therapy? And can you give me any examples of a successful cell therapy? Any takers?
People talk about stem cells.
Stem cells is actually really a really good example. And it's definitely a therapy that's up and coming as well, and that is definitely already being used, like hematopoietic stem cells are being used. But I was actually thinking of a much simpler form of cell therapy.
I hope, because of the background noise, I'm not mishearing and repeating, but I believe that it is starting to be used successfully to control degeneration of some of the receptor cells in the eyes. I think is it
the limbal cell therapy.
Yes. And the other thing is, does what you're talking about cover the use of teaching people's immune system to recognise and destroy cancer?
This is fantastic. It is a really well-educated audience. You already know quite a lot about cell therapies. I can just go home now. But there's a really simple cell therapy that is highly successful and has been used for many decades. And it's blood transfusion. Blood transfusion is a cell therapy. And it was first recorded blood transfusions took place in the 17th century. And they didn't get going until the beginning of the 20th century. And it was really driven by the First World War, when there was a real need for a lot of blood transfusions. But it's an example of a successful cell therapy. And then the stem cells, the hematopoietic stem cells, for instance, is a really good example as well. And bone marrow transplantations were first done in 1957. And that's done for people that have cancer of the bone marrow and the blood, their bone marrow is destroyed and then replaced with a healthy bone marrow, and that can be a really effective treatment. And it's also being used in children with primary immunodeficiency, where there's a gene defect in their immune cells. So these children can be treated by wiping out their bone marrow and again replacing it with bone marrow from a healthy individual, so you can restore the genetic defect. So cell therapy is already here. Now, what I also would like to say is a kind of a definition about cell therapy. You can see it as a living drug. It involves administration of whole living cells to a patient with the goal to either replace or regenerate certain tissues or cells, or to correct the faulty behavior of certain tissues and cells. Now, this brings with it its own complications, risks, technological and also ethical challenges. So I've got another question for you before I continue. What in your mind is the difference between a conventional drug therapy, think of antibiotics or aspirin? What is the difference between a conventional drug and a cell therapy? Is there anybody who would like to have a guess? Sir, sorry?
The focus,
They what?
antibiotics go for the whole body.
Yeah.
Cell therapy
Is focused?
go for one particular kind of cell.
Yeah, yes, sir.
Can I suggest an organisational difference? I may be talking nonsense. That conventional drugs are essentially static, whereas cell therapy is essentially dynamic.
That's a very nice differential you highlight there. Anybody else? Phil. Cells can grow. Oh, I hadn't written that down on my list. Yeah. Excellent. Yes, that's absolutely right. And some cells will grow and others won't. But to just also kind of highlight a few differences, I think conventional drugs really target a certain biochemical pathway. Like aspirin will block the formation of prostaglandin E2. Now, cells, because of their nature, they carry a lot of molecules and they actually are able to target multiple pathways at the same time. Now, drugs can be made in large quantities. They're available off the shelf. But cell therapy, because of limited cell availability, you need to get the cells from somewhere, can only be produced in much smaller quantities. And as a result, with conventional drug therapy, you can treat a large number of people. And with cell therapy, you may usually only treat one person and maybe a few at best. So if you kind of weigh up those things, cell therapy doesn't come off too well, I think. So why do we need cell therapies? And I'm trying to make a point that we really need them. So let's contrast two different patients. So one has a bacterial infection and another one has got an autoimmune disease. So what we mean, it's a chronic inflammatory disease where the patient's own immune cells attack their own tissues. So the joints in rheumatoid arthritis patients or insulin-producing cells in type 1 diabetes. So in the case of the bacterial infection, it's clear who the culprit is. It's the bacteria. So if you have drugs like antibiotics that were designed to kill these bacteria, if you treat the patients, you kill the bacteria, the patient is cured. It's a very linear and very successful way of treating disease. So you define a clear target, you design the killer drug, and you've got a cure. Now let's go to a patient with autoimmune disease. And then it becomes difficult because we can't draw the same parallel as with a bacterial infection. Because how do we define the culprit? It's not a well-defined bacteria, but the patient's own immune cells that cause the problem. So what do we target? How do we know who's the goodies and who's the baddies? It's not that easy. And the immune system is a very complex system. It consists of many different cell types, and these cells operate by secreting an array of proteins that can cause inflammation. So what are the options that we currently have? Well, we could wipe out the whole immune system. That would cure the disease, but it would also kill the patient because it would leave them totally unprotected to pathogens. We could weaken the immune system, we could dampen it. And yes, that's being done by using immunosuppressive drugs like steroids. And this works to an extent, but it also suppresses the beneficial immune responses that we need to thrive, and it doesn't provide us with a cure. Now, we could also be very smart and target and destroy or neutralize certain aspects of the immune system that are involved in the autoimmune attack. And yes, that has made a massive difference and has been a revolution in its own right. And it helps to fight symptoms in autoimmune patients. You may have heard of the therapeutic antibodies against TNF or deleting antibodies that delete B cells. But again, the downside of this approach is again a weakened immune system in the patients, and again, it doesn't provide us with a cure. And why not? Well, this is a very linear approach of treating patients, like for treating patients with a bacterial infection. And it just simply doesn't work because in autoimmune diseases, there's not a single culprit that is responsible, but it's a very complex network of immune cells that interact with each other, that give feedback to each other, that produce hundreds of different proteins to communicate and to orchestrate an immune attack. And so taking just one cell or protein out of the equation may lead to an improvement of symptoms, but it's unlikely to cure the condition. So why is a cell therapy different then? Well, cell therapy, it's not a linear treatment where we're trying to kill a particular culprit, but it's a treatment where we're trying to change the behavior of the immune system. So what we're trying to do is to re-educate the immune system to correct what's going wrong. So instead of punishing the immune system for doing wrong and putting it in prison and restricting it or putting it on death row, we're trying to rehabilitate it. And we believe that we can achieve this by using immune cells as a therapeutic tool, because cells are designed to communicate with other immune cells, like happens in real life, like happens in your body all the time. Cells are fully equipped to take part in the complex network of the immune system, and in this manner they can target multiple pathways and multiple cells at the same time and correct the faulty behavior of immune cells. So our story. We started to develop a cell therapy for rheumatoid arthritis now over a decade ago. And the first thing we had to consider was which cell type to choose for our therapy. As mentioned before, the immune system consists of multiple cell types, each with their own specialty. It's perhaps easiest to compare the immune system to the armed forces. Within the armed forces, there are different types of trained soldiers that can carry out specific missions. Think about infantry soldiers versus an Air Force pilot. They make use of a variety of weapons and skills to fight the enemy. And the action of these soldiers is ultimately controlled by generals who devise a strategy to combat the enemy and who control the operations carried out by the soldiers on the ground. And so for our therapy, we decided not to make use of the soldiers, but to make use of the generals of the immune system. And there's nothing wrong with aiming high, I think. And the cell type in the immune system that acts like a general is called the dendritic cell. Now, these dendritic cells circulate in the blood, but they can also be found in all our tissues, like the skin, the gut, the organs of the immune system, the lymph nodes, and also in our joints. So our basic idea was to take out dendritic cells from the blood of an RA patient, to influence the behavior of these dendritic cells in test tubes in the laboratory, so that they would become pacifistic, peacemaker generals. And then we would inject them back into the patient, where they would then instruct the soldiers of the immune system to cease fire. Easy. But like for the armed forces, the generals of the immune system are few and far between. And so we had to come up with a solution to this problem. And the solution was to train our own generals from a cell type called monocytes. They are very easy to extract from the blood. So from a pint of blood, we can get 50 million of these monocytes. And we know how to train them in test tubes in the laboratory so that they become dendritic cells. And this training process takes 7 days. Now, during their training, we ply them with drugs so that they become pacifistic generals, peacemakers, and we also give them further instruction so that they will stop the war against the patient's own tissues, but not the war against invading pathogens. Now, the drugs we use for the pacification of these dendritic cells are commonly known drugs, and you most likely have heard of them. The first one is dexamethasone. It's a glucocorticoid used in the clinic already as an immunosuppressive drug. And the other drug, well, I don't know if I should call it a drug, it's a vitamin. It's vitamin D3. Now, this is a compound that our own body makes in response to sunlight, and it's not only important for healthy bones, but also for the normal functioning of the immune system. So we use these drugs to our advantage and use them to generate dendritic cells that are equipped to act as peacemakers, that switch off the aggressive immune response to self-tissue, the joint tissue in the case of RA patients. Now, in scientific terms, we refer to these cells as tolerogenic dendritic cells. So dendritic cells that promote immune tolerance. Now, tolerogenic dendritic cells is a bit of a mouthful, so in the lab we like to call them TOL-DCs. But if you're interested in reading up about them and you want to go on the internet, then use the term tolerogenic dendritic cell. Now, like I said, the process of generating these TOL-DCs in the laboratory takes 7 days, and after this time, the cells are ready to be used as a therapy. Now, I've spoken a lot about this therapy, so I just wanted to quickly summarize how that would work for the patient. So a rheumatoid arthritis patient is coming to the clinic and they give blood. We then take their blood to a specialized laboratory facility where we extract the monocytes from the blood, and we then put these monocytes not in test tubes but in plastic culture bags. And we train the monocytes there for 7 days to become these pacifistic or peacekeeping generals or TOL-DCs. So after 7 days, the RA patient comes back to the clinic, and after we have established that the TOL-DCs conform to certain and very strict quality control standards, so we check that the product isn't contaminated with microbes, we check that the cells are alive, and we check that they look like TOL-DCs, we then inject them back into the patient. So the patient is getting their own cells, but they have been re-educated. And hopefully in the patient, they will then start to exert their peacemaking role. Now, like I said at the beginning of this talk, we have conducted a phase 1 trial in RA patients. And we have checked mainly for the safety of this approach, of this TOL-DC therapy. Now, checking for safety is always the first step on the long road of clinical trial testing. But what we also did, we checked that producing this cell therapy was feasible. So could we make a good quality TOL-DC product from all the different patients that we treated? And importantly, and not to be underestimated, we also asked the patients what they thought of the therapy. Did they think it was acceptable? Now, we got good results on all counts, which is positive. Did we cure anybody? No, we didn't. But we didn't expect that would happen, as we knew beforehand, before we started the trial, that for TOL-DC therapy to be successful, many questions remain to be answered. And this brings me to the third part and last part of this talk, in which I will try and explain some of the difficulties that we are facing with bringing this type of therapy to routine clinical practice. First of all, the use of TOL-DCs as a way of specifically switching off the aggressive autoimmune attack is completely new. We were pioneers really in this field. And so when we designed our trial, we really didn't have much to go on, and we had to do a lot of guesswork, and it educated guesswork, but nevertheless it was guesswork. So for example, we did not know how many TOL-DCs to inject, how many injections would we need, 1 injection, 10, 100, and also which injection route would be best. Should we inject intravenously into the skin, into the lymph nodes, or into the arthritic joints directly? And we thought really long and hard about all these questions. And in the end, for our trial, we decided to inject the TOL-DC into the patient's arthritic joint, knee joint. And the reasoning behind was that in this way, the TOL-DCs would be able to find the aggressive immune cells that attack the joint and switch them off. But we also know that for TOL-DCs to be truly and fully able to rehabilitate the immune system, they will also need to interact with immune cells in the lymph node, and these are the headquarters of the immune system. We hypothesized, again on lots of information we could find, we hypothesized that after injection of TOL-DCs into the joint, some would stay there to switch off the destructive immune response locally, and some would travel to headquarters, to the lymph nodes, to bring about a complete state of tolerance to joint tissues all over the body. Now, we are currently actually conducting a study to test this, and we've just done the first patient and are awaiting the next patient. So we have radio-labeled TOL-DC, so we labeled them with a radioactive label that we can see. And so we can see where they travel to after injection into the joint. Now, if our work shows that TOL-DCs indeed travel to the lymph node, then that would be absolutely fantastic, and we're all set for a phase 2 trial. But if TOL-DCs don't travel to the draining lymph node, we will probably have to go back to the research lab and improve the design of our TOL-DCs. And this would not be the end of the world. I mean, this is life, this is science, this happens all the time. And in fact, we already have good ideas how we could improve the ability of TOL-DCs to travel to the lymph nodes. But it would mean that further clinical trials would be delayed. But then again, we've already been working on this for over 12 years. A few years more is not a problem. We also still have many questions about the dosing and number of injections we need for TOL-DCs to give clinical benefit to rheumatoid arthritis patients. And in our first safety trial, we were very, very careful indeed. And we only did a single injection. We started off at very low cell numbers. We started off with 1 million, then 3 million, and then finally 10 million TOL-DCs into the knee joint. And of course, at the 10 million dose, we started to see some clinical benefit. And with hindsight, I wished we had been a bit more brave and had planned to include higher doses and multiple injections. But that's hindsight. And I do believe it was the right decision to be cautious because of the novelty of this approach and the unknown of this new treatment. Another big question we have is, at what stage of the disease will TOL-DC be most beneficial? Again, we have carefully thought about this, and we have good reason to believe that treatment with TOL-DCs will be most effective at the very early stages of rheumatoid arthritis, when the immune system is just starting to derail, but before any major damage to the joint tissue has been done. Because please note, TOL-DCs have been designed to rehabilitate the immune system, but they do not repair the damaged joint. That's another therapy. But treating patients with very early symptoms of rheumatoid arthritis brings about its own issues, and we can perhaps discuss this after this talk. One issue is, for example, if you're a rheumatoid arthritis patient with very early signs and symptoms of the disease, would you be willing to try a highly experimental cell therapy that may or may not work, knowing that their arthritis at that stage of disease can perhaps be managed quite well with currently available drugs? So that's something to consider. Finally, another obstacle I'd like to mention in getting TOL-DCs to the clinic is something that affects all different types of cell therapies, and it's the very nature of these therapies. It's the fact that they are living cells. They are extracted from individuals. So unlike antibiotics that can be mass-produced and are available off the shelf at every pharmacy in the country, a TOL-DC therapy has to be produced for every single patient that is going to be treated. This raises a number of problems. Production of TOL-DC has to take place in highly specialized facilities. There's only a few in the country that would be able to do this at this point in time. It needs to be done by specialists, highly trained staff. So it's very expensive, it's not high throughput. Every single product needs to be quality controlled. And because it's so expensive, clinical trials become very, very expensive to conduct. And it's making it very hard for us to find sufficient funding to address all the questions that we need to address, like dose, number of injections, the injection route. And this is really slowing down progress. It's a problem. And because cells can only be produced at dedicated and specialized facilities, patients who are going to be treated would need to travel. And that's another thing we need to think about. And then the final point is, because we make the product from every single patient, how can we make that reproducible? How do we know that the TOL-DC product from one patient is going to be the same as for the next patient? And how do we know the TOL-DC product we're making today is going to be the same as in 10 years' time? Because it's a human cell product, undoubtedly there's going to be variation between products. Humans are different, so the cell products we make from them are going to be different. There's going to be variation. So what we need to do is find ways to reduce the variability in the cell product. But also we need to define the limits of to what extent the variability is acceptable. And so these are all problems that come with the nature of the therapy. Having said that, I think all these problems I just mentioned are of a technical nature and we can overcome them. I think the main questions we really need to provide an answer to are, first of all, is TOL-DC therapy safe? Crucial. And secondly, if it is safe, will it provide us with a treatment that is better than any other available drug on the market today? And if we can say yes to both those questions, and if we can show that TOL-DCs may even provide a cure for autoimmune diseases, I don't think there will be a problem overcoming technical obstacles and also reducing the costs of these treatments. And so to give you an example, I think I'm not the only one who believes that cell therapies is the future. There's actually a lot of companies that believe that there is a future in cell therapies. And they have already started to develop fully automated machines that will produce cell therapies in fully closed systems. So one could envisage that blood from a patient goes into one end of the machine, and after a few days, a fully ready, pure cell therapy product comes out at the other end. And that's not science fiction, that those developments are ongoing and they're making good progress. And ultimately, such machines would make the production of cell therapies cheaper and would open up the possibility to treat patients all over the country. So I hope I've given you a flavor of what's going on in the field of cell therapies and also what we're doing here in Newcastle. And I hope I've given you some insight into the many unanswered questions and issues we are currently dealing with. Will TOL-DC therapy be available as part of routine clinical care in 50 years' time? I honestly do not know. I hope so, but I honestly do not know. But I do know that there's a lot of scope for cell therapies, and I do think that some of the cell therapies we are developing within the wider field today will lead to new treatments that will change the face of medicine. Thank you very much for your attention.
Catharien North East is supported by Newcastle University, Peels and Dance City. We're also supported by DC Cafe, who host the events.